I think I have some pretty interesting things to share with you tonight. So, as we all know, the Indian River Lagoon has had a tough year. It's been a really tough year. In the beginning of last year, in March 2016, we had a massive fish kill in the northern part of the Indian River Lagoon. And then over the summer, we had this really vile algae bloom in the southern part of the Indian River Lagoon. And it's had a tremendous impact on a lot of people's lives. Now, the data that I'm going to be talking to you about that I'm particularly proud about is the Kilroy data. So the idea was to create something that was smaller and cheaper, which is, always the direction we try to go with electronics. So it's 2/3 the cost of comparable systems. It is smaller, so it can go into canals. And it measures a whole range of things: flow speed, direction, depth, water temperature, conductivity. Blue-green algae, that's a really important one for what I'm going to be talking about, and nutrients, nitrite, nitrate, and orthophosphate. And the idea is that it provides real-time data wirelessly. It's pretty easy to install. This is what the actual unit looks like. So the sensor package is underwater. Everything's attached to that yellow pole that we call Kilroy's hat rack. And the communications box is basically just a cell phone technology to communicate to the internet. And then it's all powered by a solar panel. And the whole unit is being attached to that yellow fiberglass pole, can be installed quite rapidly. And so, as I said, we now have Kilroys up and down the Indian River Lagoon. We have 26 of them in the Indian River Lagoon. And this is kind of a dream come true for me. So a couple of years ago, we actually managed to get funding from the state, which is being administered to us through DEP. It's allowed us to put these real-time monitors out in all of the major inputs that are coming into the Indian River Lagoon. And we've made this data available to the public. If you go to our website, which is teamorca.org, and you click on our little Kilroy guy here, you can go to and see what is going on in real time at any of those sensors up and down the lagoon. And we had Kilroys in place during that fish kill in March. This was a massive fish kill. And as it happened, we had 2 Kilroys within the kill zone, which you see as the red area there, and a bunch of them in the surrounding area, which means we got the most comprehensive data set ever collected on a fish kill. And it revealed a lot of things. First of all, there was a massive brown algae bloom in the northern part of the lagoon. The brown algae died, and at that time, as it died, the microbes consuming the algae also consumed the oxygen, and at the point when the oxygen bottomed out to 0 was 3 a.m. on March 19th. The first pictures started appearing on March 20th of the dead fish. Now, this is hardly a revelation. Algae blooms are frequently responsible for fish kills for exactly this reason. But what we found in this case was that there was actually a perfect storm of events that came together to contribute to the size of this fish kill and the extent of it and the number of species that were involved. So we had heavy rains last year, which led to this massive algae bloom. The die-off of algae led to a decrease in dissolved oxygen. But there was something else that happened. In the 11 days preceding the kill, the water temperature went up an astounding 6 degrees centigrade. That's really a lot in a very short period of time. The warmer the water, the less able it is to hold on to oxygen, but it also may have favored a viral infection that might have also contributed to the die-off of the algae. Another thing that was peculiar, considering that the temperature was going up, is that we had a series of cloudy days leading up to the kill. And the day just before the kill was heavy overcast, which of course is pretty unusual for us. But that meant that respiration was favored over photosynthesis. Photosynthesis produces oxygen, but it can't do it if there's no sunlight. And so that contributed further to the decline of oxygen. And the most surprising finding was that there was a connection between a decrease in barometric pressure and an influx of anoxic groundwater. And so what happened was a storm system came through, there was a decrease in barometric pressure, and it contributed to an outflux of this anoxic water from the groundwater. And that added the final nail in the coffin. So having those Kilroys in place was incredibly useful in terms of trying to dissect what was actually going on with this fish kill. Then we had this horrendous algae bloom. For those of you that missed it, be very grateful because it was really, really disgusting. So it started in Lake Okeechobee and was carried out with all of the outflow from Lake Okeechobee. That was so much water dumped out of Lake Okeechobee that it turned the St. Lucie estuary fresh. And the alga that was responsible for this bloom is a freshwater alga. So if the estuary had been in its normal state, you wouldn't have had that bloom down there at all. But it produced this incredible amounts of what is called blue-green algae, but in fact isn't algae. It's actually a kind of photosynthetic bacteria called cyanobacteria. And cyanobacteria have been on this planet for about 3 billion years, and they are actually credited with being the source of the oxygen that you and I breathe. They were the first producers of oxygen on the planet. So it is commonly called blue-green algae, and the blue is from one of the pigments that's in it. This is the sort of thing we were seeing down in the St. Lucie estuary, where the beaches were just slimed with green. And you can imagine what it did to the property values of this multi-million-dollar home, not to mention what it did to the businesses that depend on the water, because they couldn't touch the water. Because this is a really nasty, nasty type of cyanobacteria. I mentioned it's called blue-green. Here you see the blue. So just like in the fall, up in the winter, when the leaves are turning and the chlorophyll is dying, and then you start seeing the other colors, the reds, the oranges, the yellows, that's what's happening here. The chlorophyll is dying and revealing the phycocyanin, which is the blue in the blue-green algae. It is known that this is very unhealthy for living things. Animals like farm animals and dogs that have drunk water that have this particular type of cyanobacteria in it have died. There have been some human deaths down in Brazil. A dialysis clinic, unfortunately, used some contaminated water, and they had 75 of their patients die as a consequence of it. The molecule responsible, coming from microcystis, is called microcystin, and it's quite a remarkable molecule because of its stability. You can boil this thing, and it doesn't phase it. It does not break down enzymatically. It stays around for a long time in the environment, and that's very concerning because it is extremely toxic. One drop in a tanker truck full of water would be the equivalent of 1 part per billion. That is the limit, according to the World Health Organization, of how much microcystin you should allow in drinking water. Anything over that they consider too dangerous to consume. And 10 parts per billion is the limit for recreational exposure. And what you have to understand is that even if you're just living next to this water, it's a really bad thing because it gets aerosolized. Anybody who was living down there could tell you, they knew anytime the wind changed, because in the afternoon, and there were parts of Martin County where it was just intolerable when the wind changed. Now, we're not the only ones that are suffering from this. Lake Erie in 2014 had such a massive bloom that you could actually see it from satellites from space. And it caused a tremendous problem because they started taking the water in through the Toledo water intake crib, which you see in the background there. And they actually had to shut down water service for 500,000 customers. And this was not a boil notice, which is some of us have experienced. This was a do not use notice that these people had to deal with. I'm a big believer in the crossover between art and science, and so I was very pleased when I discovered this painting, which is kind of done in the style of the murals of Diego Rivera. It's called Toledo Microcystin. And the artist, Drew Roberts, who painted it, turns out before he was an artist, he was a geologist who worked on detecting environmental contamination, which is why he gets so much right in this painting. So you see the people of Toledo having to depend on bottled water. That's the Toledo intake crib, as they call it, which all of them would recognize. But you also see him showing how microcystin is getting into the food chain, not just the fish and the bivalves, but even into the crops, because crops irrigated with microcystin can take up microcystin. And I think that that's something that we need to be very concerned about, because we've actually got a bigger problem with this than I think many of us realized. As you probably know, the history of Florida is the history of canals. Canals started to be being dug in Florida in the 1900s with the idea of making what was viewed as useless land into useful land by draining it and turning it into agricultural land. As a result, a lot of our agriculture has these canals surrounding it. And it's not only to keep the land dry for agricultural purposes, but it's also to keep it wet because they can use the water from the irrigation ditches, as they call them, to irrigate their crops. Now, hopefully, obviously, when you've got a canal that looks like this, you should not be using that water to irrigate your crops. But there's nothing to keep them from doing it. There are no laws, no regulations in place to test water that is being taken from these ditches and used for irrigation. And we found with the Kilroys that we had in the canal systems that what we saw are cryptic blooms of cyanobacteria that seem to be occurring out there a lot of the time, mostly over the summer. So when I say cryptic, I mean that they didn't produce the green slime. You didn't see any visual evidence of these blue-green algae blooms, but they're going on a lot of the time, and our crops are being irrigated with them. This is at the northern part of the lagoon, Ogali. This is over a 3-month period a couple of years ago. And Vero Shores. Queens Cove North is not a canal, and you don't see those kind of blooms. Taylor Creek, Moores Creek, both, we tend to see a lot of stuff like that coming out of there. This is from the C24 canal, one of the Kilroys that we had well up at the canal. And these concentrations are a buildup from February until the early summer. And what you're seeing here are concentrations of chlorophyll that, according to the World Health Organization, are comparable to, now we didn't actually measure the microcystin levels, which I have to make clear, but had they been producing microcystin, then we were well above just the recreational exposure limit of 10 micrograms per liter early on, and we were well into the high health effects probability by early summer. So what we found with our Kilroy data is that we have a situation in Florida that's quite different than up north, where in the northern regions where we have more structure and hilly watersheds, the nutrients run off and have a short residence time and get flushed out into lagoons or lakes where the algae blooms occur. But because Florida is so flat and because we have these weirs that block the flow in the canals, we can get major blooms in these canals. And so we have a very different situation with the algae building up in the canals. And what happens is these are freshwater algae. When they hit the brackish water in the estuary, they rupture. They release their nutrients, the dead cells, and the cyanotoxins into the muck. And this has important implications for the basin management action plans because they are looking at nutrient loads coming into the lagoon. But if they're measuring in a situation where we've got one of these algae blooms, they're going to see lower nutrients than are actually there because a lot of those nutrients are sequestered within the cyanobacteria. So I think this is of concern on a number of different fronts. Obviously, the muck that they're contributing to is a major problem for the lagoon. Any of you that spend any time in the lagoon are well familiar with this black mayonnaise that is smothering the life in the bottom of the lagoon. It gets resuspended with storms and boat traffic. It blocks out sunlight. We've got a little demonstration up here. We brought some muck that we collected, was it today? And, the stuff just stays suspended for a really long time. And it, as I said, blocks out the sunlight. It hangs on to nutrients. It hangs on to pollutants. It's a really serious problem. Another concern came from this study done out in California. This was a case of otters, sea otters, that were dying from microcystin intoxication, which was very surprising because microcystin is freshwater, and these are saltwater organisms that are eating saltwater bivalves. But as I told you, microcystin is a really tough molecule. And it turns out it's coming out of these freshwater systems, going out into the saltwater environment, invading the bivalves where it gets bioaccumulated and then being consumed by the otters, which are dying because of liver disease. So they have what are called these bloody livers that are the ultimate cause of death. But the thing is, California has some of the most stringent requirements in the country for testing for toxins in seafood, but they're not testing for freshwater toxins in saltwater seafood. So this is another major concern. And finally, as I mentioned, there's the issue of what's happening with irrigating our crops with this stuff, because it has been shown that our food crops can take up microcystin. And microcystin is, a cancer-causing agent. It causes liver cancer. So it's been shown that microcystin can be taken up by leafy plants like lettuce, spinach, cabbage. It's taken up by rice. And once again, this is not something that we need to throw at the farmers as being at fault, because I think that this is something that, everybody needs to work together on. The farmers may be impacted by it in ways that they're not realizing, because we don't have any idea how microcystin might actually be impacting the microbial community in the plant, in the soil, and the plants themselves. So their crop yield might go up if they reduce the microcystin load. So, it could end up being, better for them if we found ways to mitigate this issue of the microcystin. Microcystin we know about because it actually has both an acute and chronic effect. So I told you about the acute effects. I mean, it stinks, but it makes you feel dizzy and nauseous. And so there was an immediate connection with the source of the problem. But it also has a chronic effect in that it eventually, decades later in some cases, causes liver cancer. What about toxins that are being produced by all of these cyanobacteria besides microcystis that don't have an acute effect? We're going to find out about them later. And so this is the liver toxin from microcystin, but there's another one that concerns me greatly called BMAA, which stands for beta-methylaminoalanine. Now, this is a very interesting story. This is a very small molecule that kind of behaves like an amino acid, which is the basic building blocks of proteins, but it's not one that we normally use. Unfortunately, our body mistakes it for one that we normally use. It mistakes it for serine, builds the protein with this molecule instead, and then our proteins misfold and form those sticky plaques in the brains that are associated with Alzheimer's, Parkinson's, and Lou Gehrig's disease. Now, the connection with BMAA and these diseases came about in a very interesting way. At the end of World War II, doctors coming into Guam discovered a really bizarre situation among the Chamorro people there, where they had a combination disease that was a combination of Alzheimer's, they had the dementia, Parkinson's, they had the tremors, and ALS, they had the wasting disease of ALS and eventually would die of the muscular wasting. But it was appearing in people with ages from 20 to 72, and it was, during the 50s, the leading cause of death amongst these people. Well, needless to say, this attracted a lot of attention from the scientific and medical community. People rushed in to try to understand better what was going on. And there are a number of hypotheses, and none of them are a firm thing, but the leading hypothesis right now is that this tree, the cycad tree, produces a fruit. The cycad tree actually has a symbiotic relationship with the blue-green algae that produces BMAA, and so it accumulated in the fruit, which was then eaten by fruit bats, and then the people were eating the fruit bats. And the reason it suddenly increased as it did is that the Chamorro Indians first got guns in the 1920s. And when they started getting guns, they started shooting the fruit bats. And so they were eating fruit bats more. And now we see this decline in the disease, which is coincident or following on the extinction of the fruit bat. They hunted it to extinction. There are other kinds of data that have shown up in different places around the world linking BMAA to these diseases. there was a study that came out which preceded this article in the UK, and this study in the UK, that showed that monkeys that are given a diet made that is rich in BMAA developed Alzheimer's-like illnesses after less than 5 months. And there was a study done on, it was done on Canadians with Alzheimer's. They found that those with Alzheimer's, that died of Alzheimer's, had BMAA in their brains, and those that died of cancer did not have BMAA in their brains. Now, as a scientist, I have to tell you that this is not, first of all, correlation is not proof of anything. It's not causation. And you have to be very cautious in interpreting these things. For example, there are a group of scientists that claim that a lot of times when people think they're measuring BMAA, they're not, because it's such a small molecule, it's hard to get it right. There are definitely dissenters that don't think this is true. I have an open mind on the subject, but I would like to see more research being done on it, because I think, it's something that we ought to be paying attention to. And it does seem to be growing in public awareness. So why do we have this increase in cyanobacteria? Well, it's happening, as I mentioned, nationwide and actually worldwide, and part of the reason is climate change, because you have these cycles of drought and flood that tend to favor the productions of these cyanobacteria. But there may be another reason, as I mentioned. Now, as I said, our original goal with putting the Kilroys out there was to be able to see where the pollution is coming into the Indian River Lagoon. And I'm very proud of this next graph, and it represents the hard work of a lot of people in this room, really, really hard work. But this is a year's worth of data from the Kilroys up and down the Indian River Lagoon. So Turnbull Creek is our northernmost Kilroy. Manatee Pocket is our southernmost Kilroy, which is down here. These are all the Kilroys in between, so they're all of the inputs to the Indian River Lagoon. And you'll notice that there's kind of zones where this northern zone has very low input. This is nitrite plus nitrate, and it's the mean for a one-year period from November 1st, 2015 to November 1st, 2016. In the middle zone here, we have higher nitrite and nitrate, and then the highest regions are from the canals down here in the southern part of the lagoon. The brown in this picture is the watershed area, and we have a very large watershed in the southern part of the lagoon that is fed from these basins. So the C23 canal feeds into the North Fork of the St. Lucie River here, the C24 into the North Fork there, the North Fork itself is coming in. The C44 is, of course, the one we hear the most about, because that's feeding from Lake Okeechobee. And so if we go back and look at this again, you'll see that the highest input is from what we call the Palm City Bridge, which is our C44 canal. But right behind it is the C23 and the C24, so that's not coming from Lake Okeechobee. That's coming from those drainage basins that I just showed you. Now, that's largely agriculture, but it's old agriculture because it used to be citrus groves, and a lot of those citrus groves are fallow. We also looked at phosphate. So this is phosphate over the same period, one-year period, very low phosphate in that upper region, middle zone, kind of a mid-range. And once again, actually the Palm City Bridge is not the highest source of phosphate. C23 is the highest source of phosphate. So what does this mean in terms of the cyanobacterial blooms? Well, now I'm going to give you a little chemistry. I'm going to try to make it as painless as possible. But I think, this audience is well aware that we have issues with nitrogen and phosphorus. Nitrogen seems to be the bigger problem within the lagoon itself. But all nitrogen is not created equal, so prepare yourself. So there's three major forms that we need to be concerned about: ammonium, nitrite, and nitrate. And just to clarify the ammonium, actually ammonia is a weak base, and ammonium is a weak acid. And so which form this is in is dependent on the number of hydrogens, or what's called the pH of the water. And given the normal pH of the estuary, most of this is going to be in the form of ammonium, which is a good thing because ammonia is any of you that have aquariums know ammonia is very toxic to aquatic life. So ammonium is not so toxic, but it still may be a major problem. And the reason being is that the amount of energy in it. So when we talk about the energy in food, cells obtain energy by burning fuel, or what's also called oxidizing fuel. And that word oxidation came from the idea of oxygen binding to a chemical, because that's how we first understood oxidation. Now we know it has to do with electron transfer, but for now, let's just talk about it from the point of view of oxygen. Well, this is a fat molecule, and you can see there's no oxygen. And this is a carbohydrate molecule, and you can see there's oxygen. And what this actually means is that this has a lot more energy than this does. As we all know, right? Fat stores energy. And so all you've got to know is that when we're talking about nitrite, nitrate, and ammonium, they have different amounts of oxygen. So this one you can think of as being burned. This one's partially burned, and this one's still got all of its energy stored. There's no oxygen attached to it at all. And so you just think of them in terms of their energy levels. And what this means is that you should think about ammonium as being to nitrate as a Big Mac is to celery. Ammonium is a big energy charge. And there was a paper that just came out last year that I think is a pretty big deal by Pat Gleibert and others. And they make the case that the ratio of ammonium to nitrate actually may be the reason that we've started to see more cyanobacteria blooms. And so what they say is that the algae that we're most used to, diatoms, tend to take up nitrate preferentially over ammonium, and they store nitrate in their cells. But these little guys, the cyanobacteria, actually take up ammonium preferentially over nitrate, and they store ammonium in their cells. Dinoflagellates, some of them actually can consume other small algae, and when they do, they produce ammonium as well. So their case is that when you've got a high ratio of ammonium to nitrate, you tend to favor the picoplankton, the tiny little guys, the cyanobacteria, whereas when you have a high ratio of nitrate compared to ammonium, then you tend to favor the diatoms. And there's research to back this up when you do what's called a mesocosm study, when you mix the cells together. If you feed them nitrate, it tends to favor the production of diatoms. And if you feed them ammonium, it tends to favor the production of cyanobacteria. Interestingly, it's also been shown if you take microcystis, the one that produces microcystin, microcystis doesn't always produce microcystin, and it's never been completely clear why it does in some cases and it doesn't in others. Well, if you feed it ammonium, it's much more likely to produce microcystin than if you feed it nitrate. So that's another reason why ammonium is becoming of increasing concern. So why is there more ammonium? Well, in this Gleibert et al. Paper, they attribute it to 4 sources. The first is a shift towards the use of urea or urea-based fertilizers. And they claim that the worldwide use of urea has increased greater than 100-fold in the past 4 decades. The reason being it's cheaper. And so fertilizer companies are shifting over to the use of urea. Another source is animal waste. Animal manure comes out as ammonia. And so we've got these huge animal feedlots, and the ammonia gets gassed off and released into the environment, and it actually comes back either attached to particulates or rain as ammonium. And so animal waste, and actually human waste, just like animals, we're animals, is ammonium as well. Now, we supposedly get rid of that with our wastewater treatment plants, but only if there's tertiary treatment. So you have to be aware when you're talking about converting from septic to sewer as a solution that it's only a solution if you're willing to pay for that tertiary treatment, which is an added cost to your water treatment plant. Septic, which gets a really bad rep, maybe not undeservedly in this particular environment, actually does not produce ammonium in a properly functioning septic system. If you've got a healthy leach field that is well oxygenated, then that ammonium, before it reaches the water table, is turned to nitrate. Now, unfortunately, we have a lot of septic systems that are in non-compliance. We also have sea level rise, which is further contributing. So we do have septic systems that are part of the problem. So there is reason to think that ammonium may be a problem. There is one other source of ammonium that was not mentioned in that Gleibert paper, but I think should have been, because that is muck. Now, this is a slide produced by John Treffry up at Florida Institute of Technology. It's a slide he presented to the Florida Senate in 2013, and I think has led to some of the dredging that's now going into Brevard. But it shows the story, because this is a core sample into the bottom of the Indian River Lagoon, and you see pre-1950 we have nice clean natural sand and shell. But after 1950 we start to see this black mayonnaise, and he's managed to date it so he knows, which years this is coming from. And he's done the analysis and shows that it's 75% water, and if you've ever handled it, it's very watery stuff. It's in the solid form, it's greater than 60% silt and clay. And that's very interesting, because clay is not natural to our geology down here. There shouldn't be any clay. That clay is coming from sod. The pre-grown grass that we put on people's lawn, it's grown in clay. So all of that clay that is now part of our muck is coming from our sod. And then the organic matter, surprisingly, is a little more than 10%. People think it's all the organic matter, but it's about 10% of it. And the problem is us. There's just too many of us along the Indian River Lagoon. So in the population along the IRL has more than doubled from 743,000 to 1.6 million in the past 35 years. And that's putting a lot of stress on the environment in a whole lot of different areas. Now, this is a slide from a talk that John Treffry gave at Harbor Branch last year. And Harbor Branch has kindly made this available to the world. So if any of you are really interested in this stuff, I tell you, it's a really excellent lecture. And he's shown by measurements that he's made just how much ammonium is actually coming out of the muck. And the numbers are staggering. And basically, he's found that 40% of the nitrogen that is coming into the northern part of the Indian River Lagoon is coming out of the muck. I mean, more than actually from the runoff itself. Now, that's very interesting because, okay, so we've got all of these nutrients in the northern part of the lagoon, but remember that graph I showed you, we showed low nutrients coming in from the canal systems. So there actually is high nutrient up there, but it's not coming out of the canals. It's actually in the muck. And the muck up there is very deep. There's places it's 20 feet deep because there's very low turnover of the water up there. It just stays forever. And it's become a major source of nutrient, which explains why when we saw this data, we were initially a little confused by it. This is the nitrate again that I showed you earlier. But this is the chlorophyll. Well, remember, we had the big brown tide up there last spring, and this is showing that, but it's feeding off of the nutrients in the muck. So muck is a big deal. Now, we've actually been interested in muck for quite some time, and we started because when we were first developing the Kilroys, I was being funded by a foundation. I went up to make a presentation to their board, and one of their board members asked me, so how do you know where to put the Kilroys? And I said, well, you want to look at communities and, factory farms and golf courses and, try to figure out where a good place to put them was. But I thought about it more afterwards, and I thought it's still a really good question. And so I thought, well, if we look at the accumulation of muck in the pollution sinks in the sediment, that actually might be a good indicator. And it turned out that's pretty easy to do. Very easy to take sediment samples. And we've been doing that to create these pollution maps, some of which you see up here on the stage. This is one of the early ones we did. And it was a great way to look at where pollution is accumulating. So this is just like a weather map where red is hot and blue is cold, only red is very high concentrations of nitrogen and blue is low concentrations. And it shows where the problems are and actually where the solutions are, because this is the Vero Beach Country Club. It's a golf course, which often gets blamed for being a source of nutrients, but they use very low-dose organic liquid fertilizer. They bag all of their grass clippings, and they don't poison their water hazards. So it shows that there are potential solutions out there. We then applied that to the C-24 canal. We put Kilroy's at the mouth and at the transition between ag and urban. We just thought this was a pretty ideal place. We were kind of thinking we were going to find some smoking guns, but what we found is what everybody's finding everywhere. It's a whole combination of factors everywhere we look. So we did 5 seasons of muck mapping, thinking that it would change seasonally. But what we found was that the pollution was scattered throughout the canal most of the time. The average muck depth was 20 inches and appeared to contain a legacy load of pollutants. When we weren't finding sources from our muck mapping in the canal, we went out into the project culverts, and I really credit Team ORCA with this because I didn't do it, but some of these people were out there climbing through these project culverts. Anybody of you that have been out there know what we're talking about. There's snakes, there's alligators. It's a really tough slog. But they managed to get data, and it revealed a wide range of pollution that was related, not surprisingly, to the drainage area and the land use. One of the things that came out of that study that I think is worth sharing with the public is that we are concerned about the citrus groves that are being converted from citrus to row crops, because row crops are very nutrient-intensive crops. And so we have a lot of companies coming in, buying up our old citrus groves, wanting to turn them into row crops. You know, we all want to eat vegetables. Let's not vilify them, but let's try to work ahead of the game this time and make sure that this doesn't happen again so we can prevent the kind of pollution that now we're still dealing with years after the citrus has shut down. And we need to encourage them to change the Basin Management Action Plan because it turns out that the land use is very outdated, and we need to update that if the Basin Management Action Plan is going to work. We found evidence of mammalian fecal contributions in both the ag and the urban. In ag, it seemed to be cow. In urban, it seemed to be human. But as again, we've got these kind of problems everywhere. Now, we've also done these maps at places like, this is the moorings in Vero Beach, and it pretty clearly shows where you've got serious problem. The moorings are pretty typical, the landscaping that's happened in Florida, where you've got these hardened seawalls and sloping grass lawns coming right down to the edge of the seawall. It isn't just Vero Beach, this is down from the St. Lucie County area. And you can see that, you know, when you're cutting your grass, when you're fertilizing your lawns, all of that is going to be sliding into the water and creating these problems for all of us. And a lot of communities have gotten smart about this. In Virginia, they're starting to encourage people to have buffer zones and mulched beds that are planted with deep-rooted native plants. I love this from the Maine government site with the happy face. This is good landscaping. This is bad landscaping. This is Maine, where they even get into different zone tolerance for different types of plants. Michigan lakes, where they show, you know, this is bad with the grass right up to the edge, and there you've got a buffer zone. Actually, what our data is showing is that living shorelines really do make a huge difference wherever we can find them. And I've started to realize that they actually make a bigger difference than I originally thought, and I think what's happening is you probably have a successional change that is being interrupted. So nature is really good at extracting energy, and it probably starts out as ammonium and then gets gradually converted to nitrate and reduced more and more as it comes out through a natural system, and we're interrupting that with the seawall. Whereas, you know, it would be much better if we had these berms with deep-rooted plants. It's really important that they be deep roots so that the water can be collected by those roots. And we also have found, you know, much healthier muck in front of places where they have mangroves. So you can get dispensations if you've got mangroves to be able to trim them so it doesn't ruin your view. It can actually be quite pretty, but it also has the added benefit of erosion protection. Also, I go back to this map, you know, I pointed out this bad area. This is actually the moorings golf course. There's a bad pocket right there, which we think it might actually be coming from the fields right behind it. But around the golf course, it's surrounded by mangroves. So we think living shorelines make a big difference. And we have a living shorelines display up here. We're actually hoping to be working with the Indian River Land Trust on a joint project to look at the impact of living shorelines. Erosion protection, we're hoping, might actually be a way to work with the insurance industry. So if you have a seawall, it's much more prone to destruction after a storm than a living shoreline. And so when people have to replace their seawalls, if they could be incentivized by the insurance industry, give them a break, if they put in a living shoreline, that would be a win-win. Certainly there's places where that can't be done, where we have these canals that are so narrow, if you put in a living shoreline, you eliminate the canal. But we found that there are alternatives. This is benefits of living shorelines. Obviously there are many, including a lot more leisure time. You're not taking care of your lawn, you're saving it, habitat for birds and butterflies. This is a property that we found that was very clean in front of it, and it has it has these xeriscaping. It's really pretty, and it seems to work. So the point is that we need to stop pointing fingers at each other about pollution. That gets us nowhere. It's the classic tragedy of the commons, where I'm not going to stop what I'm doing because he's doing worse than I am. And so nothing gets done. What we need to do is work together to try to push that needle back towards the clean side, because otherwise the fingers that are going to be pointed at us are the next generation. The greatest threat to our planet is the belief that someone else will save it. And I thank you all for coming here and being people that care about saving it.